Chapter 26 Seed Plants 26.1 Evolution of seed plants. By the end of this section, you will be able to do the following. Describe the two major innovations that allowed seed plants to reproduce in the absence of water. Explain when seed plants first appeared and when gymnosperms became the dominant plant group. Discuss the purpose of pollen grains and seeds. Describe the significance of angiosperms bearing both flowers and fruit. Figure 26.1 Seed plants dominate the landscape and play an enormous and integral role in the success of all human societies. Here are a few examples. A. Palm trees grow along the shoreline, serving numerous purposes for food, shelter, and even transportation. B. Wheat is an important crop grown throughout most of the world. C. The fruit of the cotton plant produces fibers that are woven into fabric. D. The potent alkaloids of the beautiful opium poppy have long influenced human life both as a medicinal remedy and as a dangerously addictive drug. Credit A. Modification of work by Ryan Cozy. Credit B. Modification of work by Stephen Osmus. Credit C. Modification of work by David Nance. Credit D. Modification of work by Jolly Janner. Introduction. The lush palms on tropical shorelines do not depend on water for the dispersal of their pollen, fertilization, or the survival of the zygote, unlike mosses, liverworts, and ferns living within the same terrain. These palms are seed plants, which have broken free from the need to rely on water for their reproductive needs. The seed plants play an integral role in all aspects of life on the planet, shaping the physical terrain, influencing the climate, and maintaining life as we know it. For millennia, human societies have depended on seed plants for nutrition and medicinal compounds. Somewhat more recently, seed plants have served as a source of manufactured products such as timber and paper, dyes, and textiles. As an example, multiple uses have been found for each of the plants shown above. Palms provide materials including rattans, oils, and dates. Grains like wheat are grown to feed both human and animal populations or fermented to produce alcoholic beverages. The fruit of the cotton flower is harvested as a bowl, with its fibers transformed into clothing or pulp for paper. The showy opium poppy is valued both as an ornamental flower and as a source of potent opiate compounds. Chapter outline 26.1 Evolution of seed plants 26.2 Gymnosperms 26.3 Angiosperms 26.4 The role of seed plants. The first plants to colonize land were most likely related to the ancestors of modern-day mosses, bryophytes, which are thought to have appeared about 500 million years ago. They were followed by liverworts, also bryophytes, and primitive vascular plants, the pterophytes, from which modern ferns are descended. The life cycle of bryophytes and pterophytes is characterized by the alternation of generations, which is also exhibited in the gymnosperms and angiosperms. However, what sets bryophytes and pterophytes apart from gymnosperms and angiosperms is their reproductive requirement for water. The completion of the bryophyte and pterophyte life cycle requires water because the male gametophyte releases flagellated sperm, which must swim to reach and fertilize the female gamete or egg. After fertilization, the zygote undergoes cellular division and grows into a diploid sporophyte, which in turn will form sporangia or spore vessels. In the sporangia, mother cells undergo meiosis and produce the haploid spores. Release of spores in a suitable environment will lead to germination and a new generation of gametophytes. In seed plants, the evolutionary trend led to a dominant sporophyte generation accompanied by a corresponding reduction in the size of the gametophyte from a conspicuous structure to a microscopic cluster of cells enclosed in the tissues of the sporophyte. Whereas lower vascular plants, such as club mosses and ferns, are mostly homosporous, producing only one type of spore, all seed plants, or spermatophytes, are heterosporous, producing two types of spores: megaspores, female, and microspores, male. Megaspores develop into female gametophytes that produce eggs, and microspores mature into male gametophytes that generate sperm. Because the gametophytes mature within the spores, they are not free-living, as are the gametophytes of other seedless vascular plants. Ancestral heterosporous seedless plants, represented by modern-day plants such as the spike moss Selaginella, are seen as the evolutionary forerunners of seed plants. In the life cycle of Selaginella, both male and female sporangia develop within the same stem like Strobilus. In each male sporangium, multiple microspores are produced by meiosis. Each microspore produces a small antheridium contained within a spore case. As it develops, it is released from the strobilus, and a number of flagellated sperm are produced that then leave the spore case. In the female sporangium, a single megaspore mother cell undergoes meiosis to produce 4 megaspores. Gametophytes develop within each megaspore, consisting of a mass of tissue that will later nourish the embryo and a few archegonia. The female gametophyte may remain within remnants of the spore wall in the megasporangium until after fertilization has occurred and the embryo begins to develop. This combination of an embryo and nutritional cells is a little different from the organization of a seed, since the nutritive endosperm in a seed is formed from a single cell rather than multiple cells. Both seeds and pollen distinguish seed plants from seedless vascular plants. These innovative structures allowed seed plants to reduce or eliminate their dependence on water for gamete fertilization and development of the embryo, and to conquer dry land. Pollen grains are male gametophytes, which contain the sperm, gametes, of the plant. The small, haploid 1N cells are encased in a protective coat that prevents desiccation, drying out, and mechanical damage. Pollen grains can travel far from their original sporophyte, spreading the plant's genes. Seeds offer the embryo protection, nourishment, and a mechanism to maintain dormancy for tens or even thousands of years, ensuring that germination can occur when growth conditions are optimal. Seeds therefore allow plants to disperse the next generation through both space and time. With such evolutionary advantages, seed plants have become the most successful and familiar group of plants. Both adaptations expanded the colonization of land begun by the bryophytes and their ancestors. Fossils place the earliest distinct seed plants at about 350 million years ago. The first reliable record of gymnosperms dates their appearance to the Pennsylvanian period, about 319 million years ago, figure 26.2. Gymnosperms were preceded by pro-gymnosperms, the first naked seed plants, which arose about 380 million years ago. Pro-gymnosperms were a transitional group of plants that superficially resembled conifers, cone-bearers, because they produced wood from the secondary growth of the vascular tissues. However, they still reproduced like ferns, releasing spores into the environment. At least some species were heterosporous. Pro-gymnosperms, like the extinct Archaeopteryx, not to be confused with the ancient bird Archaeopteryx, dominated the forests of the late Devonian period. However, by the early Triassic, circa 240 Maya, and middle Jurassic, circa 205 Maya, Mesozoic era, the landscape was dominated by the true gymnosperms. Angiosperms surpassed gymnosperms by the middle of the Cretaceous, circa 100 Maya, in the late Mesozoic era, and today 670. Chapter 26, Seed Plants Access for free at OpenStax.org. Are the most abundant and biologically diverse plant group in most terrestrial biomes. Figure 26.2 Plant Timeline. Various plant species evolved in different eras. Credit: United States Geological Survey, figure modified from source. Evolution of gymnosperms. The fossil plant Elkinsia polymorpha, a seed fern, from the Devonian period, about 400 million years ago, is considered the earliest seed plant known to date. Seed ferns, figure 26.3, produced their seeds along their branches, in structures called cupules that enclosed and protected the ovule, the female gametophyte and associated tissues, which develops into a seed upon fertilization. Seed plants resembling modern tree ferns became more numerous and diverse in the coal swamps of the Carboniferous period. Figure 26.3 Seed fern leaf. This fossilized leaf is from Glossopteris, a seed fern that thrived during the Permian Age, 290–240 million years ago. Credit: D. L. Schmidt, USGS. Fossil records indicate the first gymnosperms, pro-gymnosperms, most likely originated in the Paleozoic era, during the Middle Devonian period, about 390 million years ago. The previous Mississippian and Pennsylvanian periods were wet and dominated by giant fern trees. But the following Permian period was dry, which gave a reproductive edge to seed plants, which are better adapted to survive dry spells. The ginkgos, a group of gymnosperms with only one surviving species, the Ginkgo biloba, were the first gymnosperms to appear during the Lower Jurassic. Gymnosperms expanded in the Mesozoic era, about 240 million years ago, supplanting ferns in the landscape, and reaching their greatest diversity during this time. The Jurassic period was as much the age of the cycads, palm tree-like gymnosperms, as the age of the dinosaurs. Ginkgos and the more familiar conifers also dotted the landscape. Although angiosperms, flowering plants, are the major form of plant life in most 26.1 evolution of seed plants 671 biomes, gymnosperms still dominate some ecosystems, such as the taiga, boreal forests, and the alpine forests at higher mountain elevations, figure 26.4, because of their adaptation to cold and dry growth conditions. Figure 26.4 conifers. This boreal forest, taiga, has low-lying plants and conifer trees. Credit. L. B. Brubaker, Noah, Seeds and Pollen as an evolutionary adaptation to dry land. Bryophyte in fern spores are haploid cells dependent on moisture for rapid development of multicellular gametophytes. In the seed plants, the female gametophyte consists of just a few cells, the egg and some supportive cells, including the endosperm-producing cell that will support the growth of the embryo. After fertilization of the egg, the diploid zygote produces an embryo that will grow into the sporophyte when the seed germinates. Storage tissue to sustain growth of the embryo and a protective coat give seeds their superior evolutionary advantage. Several layers of hardened tissue prevent desiccation, and free the embryo from the need for a constant supply of water. Furthermore, seeds remain in a state of dormancy, induced by desiccation and the hormone abscisic acid, until conditions for growth become favorable. Whether blown by the wind, floating on water, or carried away by animals, seeds are scattered in an expanding geographic range, thus avoiding competition with the parent plant. Pollen grains, figure 26.5, are male gametophytes containing just a few cells and are distributed by wind, water, or an animal pollinator. The whole structure is protected from desiccation and can reach the female organs without depending on water. After reaching a female gametophyte, the pollen grain grows a tube that will deliver a male nucleus to the egg cell. The sperm of modern gymnosperms and all angiosperms lack flagella, but in cycads, ginkgo, and other primitive gymnosperms, the sperm are still motile, and use flagella to swim to the female gamete. However, they are delivered to the female gametophyte enclosed in a pollen grain. The pollen grows or is taken into a fertilization chamber, where the motile sperm are released and swim a short distance to an egg. 672 Chapter 26, Seed Plants Access for Free at OpenStax.org. Figure 26.5 Pollen Fossils. This fossilized pollen is from a buckbean fen core found in Yellowstone National Park, Wyoming. The pollen is magnified 1,054 times. Credit. R. G. Baker, USGS. Scale bar data from Matt Russell. Evolution of angiosperms. The roughly 200 million years between the appearance of the gymnosperms and the flowering plants gives us some appreciation for the evolutionary experimentation that ultimately produced flowers and fruit. Angiosperms, seed in a vessel, produce a flower containing male and/or female reproductive structures. Fossil evidence, figure 26.6, indicates that flowering plants first appeared about 125 million years ago in the Lower Cretaceous, late in the Mesozoic era, and were rapidly diversifying by about 100 million years ago in the Middle Cretaceous. Earlier traces of angiosperms are scarce. Fossilized pollen recovered from Jurassic geological material has been attributed to angiosperms. A few early Cretaceous rocks show clear imprints of leaves resembling angiosperm leaves. By the mid-Cretaceous, a staggering number of diverse flowering plants crowd the fossil record. The same geological period is also marked by the appearance of many modern groups of insects, suggesting that pollinating insects played a key role in the evolution of flowering plants. New data in comparative genomics and paleobotany, the study of ancient plants, have shed some light on the evolution of angiosperms. Although the angiosperms appeared after the gymnosperms, they are probably not derived from gymnosperm ancestors. Instead, the angiosperms form a sister clade, a species and its descendants, that developed in parallel with the gymnosperms. The two innovative structures of flowers and fruit represent an improved reproductive strategy that served to protect the embryo, while increasing genetic variability and range. There is no current consensus on the origin of the angiosperms. Paleobotanists debate whether angiosperms evolved from small woody bushes, or were related to the ancestors of tropical grasses. Both views draw support from cladistics, and the so-called woody magnoliid hypothesis, which proposes that the early ancestors of angiosperms were shrubs like modern magnolia, also offers molecular biological evidence. The most primitive living angiosperm is considered to be Amborella trichopoda, a small plant native to the rainforest of New Caledonia, an island in the South Pacific. Analysis of the genome of a trichopoda has shown that it is related to all existing flowering plants and belongs to the oldest confirmed branch of the angiosperm family tree. The nuclear genome shows evidence of an ancient whole genome duplication. The mitochondrial genome is large and multichromosomal, containing elements from the mitochondrial genomes of several other species, including algae and a moss. A few other angiosperm groups, called basal angiosperms, are viewed as having ancestral traits because they branched off early from the phylogenetic tree. Most modern angiosperms are classified as either monocots or eudicots, based on the structure of their leaves and embryos. Basal 26.1, Evolution of seed plants. 673 angiosperms, such as water lilies, are considered more ancestral in nature because they share morphological traits with both monocots and eudicots. Figure 26.6 Ficus imprint. This leaf imprint shows a Ficus speciosissima, an angiosperm that flourished during the Cretaceous period. Credit. W. T. Lee, USGS, Flowers and Fruits. As an evolutionary adaptation, angiosperms produce their gametes in separate organs, which are usually housed in a flower. Both fertilization and embryo development take place inside an anatomical structure that provides a stable system of sexual reproduction largely sheltered from environmental fluctuations. With about 300,000 species, flowering plants are the most diverse phylum on Earth after insects, which number about 1,200,000 species. Flowers come in a bewildering array of sizes, shapes, colors, smells, and arrangements. Most flowers have a mutualistic pollinator, with the distinctive features of flowers reflecting the nature of the pollination agent. The relationship between pollinator and flower characteristics is one of the great examples of coevolution. Following fertilization of the egg, the ovule grows into a seed. The surrounding tissues of the ovary thicken, developing into a fruit that will protect the seed and often ensure its dispersal over a wide geographic range. Not all fruits develop completely from an ovary. Such false fruits, or pseudocarps, develop from tissues adjacent to the ovary. Like flowers, fruit can vary tremendously in appearance, size, smell, and taste. Tomatoes, green peppers, corn, and avocados are all examples of fruits. Along with pollen and seeds, fruits also act as agents of dispersal. Some may be carried away by the wind. Many attract animals that will eat the fruit and pass the seeds through their digestive systems, then deposit the seeds in another location. Cockleburrs are covered with stiff, hooked spines that can hook into fur, or clothing, and hitch a ride on an animal for long distances. The cockleburrs that clung to the velvet trousers of an enterprising Swiss hiker, George de Mestral, inspired his invention of the loop and hook fastener he named Velcro. Evolution connection building phylogenetic trees with analysis of DNA sequence alignments. All living organisms display patterns of relationships derived from their evolutionary history. Phylogeny is the science that describes the relative connections between organisms, in terms of ancestral and descendant species. Phylogenetic trees, such as the plant evolutionary history shown in Figure 26.7, are tree-like branching diagrams that depict these relationships. Species are found at the tips of the branches. Each branching point, called a node, is the point at which a single taxonomic group, taxon, such as a species, separates into two or more species. 674 Chapter 26, Seed Plants Access for Free at OpenStax.org. Figure 26.7 Plant Phylogeny. This phylogenetic tree shows the evolutionary relationships of plants. Phylogenetic trees have been built to describe the relationships between species since the first sketch of a tree that appeared in Darwin's Origin of Species. Traditional methods involve comparison of homologous anatomical structures and embryonic development, assuming that closely related organisms share anatomical features that emerge during embryo development. Some traits that disappear in the adult are present in the embryo. For example, an early human embryo has a postanal tail, as do all members of the phylum Chordata. The study of fossil records shows the intermediate stages that link an ancestral form to its descendants. However, many of the approaches to classification based on the fossil record alone are imprecise and lend themselves to multiple interpretations. As the tools of molecular biology and computational analysis have been developed and perfected in recent years, a new generation of tree building methods has taken shape. The key assumption is that genes for essential proteins or RNA structures, such as the ribosomal RNAs, are inherently conserved because mutations, changes in the DNA sequence, could possibly compromise the survival of the organism. DNA from minute samples of living organisms or fossils can be amplified by polymerase chain reaction, PCR, and sequenced, targeting the regions of the genome that are most likely to be conserved between species. The genes encoding the 18S ribosomal RNA from the small subunit and plastid genes are frequently chosen for DNA alignment analysis. Once the sequences of interest are obtained, they are compared with existing sequences in databases such as GenBank, which is maintained by the National Center for Biotechnology Information. A number of computational tools are available to align and analyze sequences. Sophisticated computer analysis programs determine the percentage of sequence identity or homology. Sequence homology can be used to estimate the evolutionary distance between two DNA sequences and reflect the time elapsed since the genes separated from a common ancestor. Molecular analysis has revolutionized phylogenetic trees. In some cases, prior results from morphological studies have been confirmed, for example, confirming Amborella trichopita as the most primitive angiosperm known. However, some groups and relationships have been rearranged as a result of DNA analysis. 26.2 Gymnosperms. By the end of this section, you will be able to do the following: discuss the type of seeds produced by gymnosperms, as well as other characteristics of gymnosperms, identify the geological era dominated by the gymnosperms and describe the conditions to which they were adapted, list the 4 groups of modern-day gymnosperms and provide examples of each, describe the life cycle of a typical gymnosperm. Gymnosperms, meaning naked seeds, are a diverse group of seed plants. According to the anthophyte hypothesis, the angiosperms are a sister group of one group of gymnosperms, the nettales, which makes the gymnosperms a paraphyletic group. Paraphyletic groups are those in which not all descendants of a single common ancestor are included in the group. However, the netifer hypothesis suggests that the nettophytes are sister to the conifers, making the gymnosperms monophyletic and sister to the angiosperms. Further molecular and anatomical studies may clarify these relationships. Characteristics of the gymnosperms include naked seeds, separate female and male gametophytes, pollen cones and ovulate cones, pollination by wind and insects, and tracheids, which transport water and solutes in the vascular system. Gymnosperm seeds are not enclosed in an ovary. Rather, they are only partially sheltered by modified leaves called sporophylls. You may recall the term strobilus, plural equals strobili, describes a tight arrangement of sporophylls around a central stalk, as seen in pine cones. Some seeds are enveloped by sporophyte tissues upon maturation. The layer of sporophyte tissue that surrounds the megasporangium, and later, the embryo, is called the integument. Gymnosperms were the dominant phylum in the Mesozoic era. They are adapted to live where fresh water is scarce during part of the year, or in the nitrogen-poor soil of a bog. Therefore, they are still the prominent phylum in the coniferous biome or taiga, where the evergreen conifers have a selective advantage in cold and dry weather. Evergreen conifers continue low levels of photosynthesis during the cold months, and are ready to take advantage of the first sunny days of spring. One disadvantage is that conifers are more susceptible than deciduous trees to leaf infestations because most conifers do not lose their leaves all at once. They cannot, therefore, shed parasites and restart with a fresh supply of leaves in spring. The life cycle of a gymnosperm involves alternation of generations, with a dominant sporophyte in which reduced male and female gametophytes reside. All gymnosperms are heterosporous. The male and female reproductive organs can form in cones or strobili. Male and female sporangia are produced either on the same plant, described as monoecious, one home, or bisexual, or on separate plants, referred to as dioecious, two homes, or unisexual, plants. The life cycle of a conifer will serve as our example of reproduction in gymnosperms. Life cycle of a conifer. Pine trees are conifers, coniferous equals cone-bearing, and carry both male and female sporophylls on the same mature sporophyte. Therefore, they are monoecious plants. Like all gymnosperms, pines are heterosporous and generate two different types of spores, male microspores and female megaspores. Male and female spores develop in different strobili, with small male cones and larger female cones. In the male cones, or staminate cones, the microsporocytes undergo meiosis and the resultant haploid microspores give rise to male gametophytes or pollen grains by mitosis. Each pollen grain consists of just a few haploid cells enclosed in a tough wall reinforced with sporopollenin. In the spring, large amounts of yellow pollen are released and carried by the wind. Some gametophytes will land on a female cone. Pollination is defined as the initiation of pollen tube growth. The pollen tube develops slowly, and the generative cell in the pollen grain produces two haploid sperm or generative nuclei by mitosis. At fertilization, one of the haploid sperm nuclei will unite with the haploid nucleus of an egg cell. Female cones, or ovulate cones, contain 2 ovules per scale. Each ovule has a narrow passage that opens near the base of the sporophyll. This passage is the micropyle, through which a pollen tube will later grow. One megaspore mother cell, or megasporocyte, undergoes meiosis in each ovule. 3 of the 4 cells break down. Only a single surviving cell will develop into a female multicellular gametophyte, which encloses archegonia. An archegonium is a reproductive organ that contains a single large egg. As the female gametophyte begins to develop, a sticky pollination drop traps wind-blown pollen grains near the opening of the micropyle. A pollen tube is formed and grows toward the developing gametophyte. One of the generative or sperm nuclei from the pollen tube will enter the egg and fuse with the egg nucleus as the egg matures. Upon fertilization, the diploid egg will give rise to the embryo, which is enclosed in a seed coat of tissue from the parent plant. Although several eggs may be formed and even fertilized, there is usually a single surviving embryo in each ovule. Fertilization and seed development is a long process in pine trees. It may take up to 2 years after pollination. The seed that is formed contains 3 generations of 676. Chapter 26. Seed plants access for free at openstax.org. Tissues. The seed coat that originates from the sporophyte tissue, the gametophyte tissue that will provide nutrients, and the embryo itself. Figure 26.8 illustrates the life cycle of a conifer. The sporophyte, 2N, phase is the longest phase in the life of a gymnosperm. The gametophytes, 1N, produced by microspores and megaspores, are reduced in size. It may take more than a year between pollination and fertilization while the pollen tube grows towards the growing female. Gametophyte, 1N, which develops from a single megaspore. The slow growth of the pollen tube allows the female gametophyte time to produce eggs, 1N. Visual connection. Figure 26.8 Conifer life cycle. This image shows the life cycle of a conifer. Pollen from male cones blows up into upper branches, where it fertilizes female cones. The megaspore shown in the image develops into the female gametophyte as the pollen tube slowly grows toward it, eventually fusing with the egg and delivering a male nucleus, which combines with the female nucleus of the mature egg. At what stage does the diploid zygote form? A. When the female cone begins to bud from the tree. B. At fertilization. Circa when the seeds drop from the tree. D. When the pollen tube begins to grow. Link to learning. Watch this video to see the process of seed production in gymnosperms. Click to view content, https://www.openstax.org/l/gymnosperm2 Diversity of gymnosperms. Modern gymnosperms are classified into 4 phyla. Coniferophyta, Cycadophyta, and Ginkgophyta are similar in their pattern of seed development and also in their production of secondary cambium, cells that generate the vascular system of the trunk or stem and are partially specialized for water transportation. However, the 3 phyla are not closely related phylogenetically to 26.2, gymnosperms 677 each other. Gnetophyta are considered the closest group to angiosperms because they produce true xylem tissue, with vessels as well as the tracheids found in the rest of the gymnosperms. It is possible that vessel elements arose independently in the 2 groups. Conifers, Coniferophyta. Conifers are the dominant phylum of gymnosperms, with the greatest variety of species. Figure 26.9. Typical conifers are tall trees that bear scale-like or needle-like leaves. Water evaporation from leaves is reduced by their narrow shape and a thick cuticle. Snow easily slides off needle-shaped leaves, keeping the snowload light, thus reducing broken branches. Such adaptations to cold and dry weather explain the predominance of conifers at high altitudes and in cold climates. Conifers include familiar evergreen trees such as pines, spruces, firs, cedars, sequoias, and yews. A few species are deciduous and lose their leaves in fall. The bald cypress, Don redwood, European larch and the tamarack, figure 26.9c, are examples of deciduous conifers. Many coniferous trees are harvested for paper pulp and timber. The wood of conifers is more primitive than the wood of angiosperms. It contains tracheids, but no vessel elements, and is therefore referred to as soft wood. Figure 26.9 Conifers. Conifers are the dominant form of vegetation in cold or arid environments and at high altitudes. Shown here are the A. Evergreen spruce Picea sp. B. Juniper Juniperus sp. C. Coastal redwood or Sequoia Sequoia sempervirens, and D. the tamarack Larix larsinia. Notice the deciduous yellow leaves of the tamarack. Credit A. Modification of work by Rosendahl. Credit B. Modification of work by Alan Levine. Credit C. Modification of work by Wendy McCormick. Credit D. Modification of work by Mickey Zieliman. Cycads thrive in mild climates, and are often mistaken for palms because of the shape of their large, compound leaves. Cycads bear large strobili or cones, figure 26.10, and may be pollinated by beetles rather than wind, which is unusual for a gymnosperm. Large cycads dominated the landscape during the age of dinosaurs in the Mesozoic, but only 100 or so smaller species persisted to modern times. They face possible extinction, and several species are protected through international conventions. Because of their attractive shape, they are often used as ornamental plants in gardens in the tropics and subtropics. 678 Chapter 26, Seed Plants Access for Free at OpenStax.org. Figure 26.10 Cycad. This cycad, Encephalotos ferox, has large cones and broad, fern-like leaves. Credit. Wendy Cutler. Ginkgophytes. The single surviving species of the Ginkgophytes group is Ginkgo biloba, figure 26.11. Its fan-shaped leaves, unique among seed plants because they feature a dichotomous venation pattern, turn yellow in autumn and fall from the tree. For centuries, G. biloba was cultivated by Chinese Buddhist monks in monasteries, which ensured its preservation. It is planted in public spaces because it is unusually resistant to pollution. Male and female organs are produced on separate plants. Typically, gardeners plant only male trees because the seeds produced by the female plant have an off-putting smell of rancid butter. Figure 26.11 Ginkgo. This plate from the 1870 book Flora Japonica, Sectio Prima, Tafelband, depicts the leaves and fruit of Ginkgo biloba, as drawn by Philipp Franz von Siebold and Joseph Gerhard Zuccarini. Netophytes. The phylogenetic position of the netophytes is not currently resolved. Their possession of vessel elements suggests they are the closest relative to modern angiosperms. However, molecular analysis places them closer to the conifers. The three living genera are quite dissimilar: Ephedra, Netum, and Welwitschia, figure 26.12, which may indicate that the group is not monophyletic. Like angiosperms, they have broad leaves. Ephedra, figure 26.12a, occurs in dry areas of the west coast of the United States and 26.2, Gymnosperms 679 Mexico. Ephedra's small, scale-like leaves are the source of the compound ephedrine, which is used in medicine as a potent decongestant. Because ephedrine is similar to amphetamines, both in chemical structure and neurological effects, its use is restricted to prescription drugs. Netum species, figure 26.12b, are found in some parts of Africa, South America, and Southeast Asia, and include trees, shrubs and vines. Welwitschia, figure 26.12c, is found in the Namib Desert, and is possibly the oddest member of the group. It produces only 2 leaves, which grow continuously throughout the life of the plant. Some plants are hundreds of years old. Like the ginkgos, Welwitschia produces male and female gametes on separate plants. Figure 26.12, a. Ephedra viridis, known by the common name Mormon tea, grows on the west coast of the United States and Mexico. B. Netum nemen grows in Malaysia. C. The large Welwitschia mirabilis can be found in the Namibian desert. Credit A. Modification of work by USDA. Credit B. Modification of work by Malcolm Manners. Credit C. Modification of work by Derek Keats. Link to learning. Watch this BBC video describing the amazing strangeness of Welwitschia. Click to view content, https://www.openstax.org/l/Welwitschia2 . 26.3 Angiosperms. By the end of this section, you will be able to do the following. Explain why angiosperms are the dominant form of plant life in most terrestrial ecosystems. Describe the main parts of a flower and their functions. Detail the life cycle of a typical gymnosperm and angiosperm. Discuss the similarities and differences between the two main groups of flowering plants from their humble and still obscure beginning during the early Jurassic period. The angiosperms, or flowering plants, have evolved to dominate most terrestrial ecosystems. Figure 26.13. With more than 300,000 species, the angiosperm phylum, Anthophyta, is second only to insects in terms of diversification. Figure 26.13 Flowers. These flowers grow in a botanical garden border in Bellevue, Washington. Flowering plants dominate terrestrial landscapes. The vivid colors of flowers and enticing fragrance of flowers are adaptations to pollination by animals like insects, birds, and bats. Credit 680. Chapter 26, Seed Plants. Access for free at openstax.org. Miriam Feldman. The success of angiosperms is due to two novel reproductive structures, flowers and fruits. The function of the flower is to ensure pollination, often by arthropods, as well as to protect a developing embryo. The colors and patterns on flowers offer specific signals to many pollinating insects or birds and bats that have co-evolved with them. For example, some patterns are visible only in the ultraviolet range of light, which can be seen by arthropod pollinators. For some pollinators, flowers advertise themselves as a reliable source of nectar. Flower scent also helps to select its pollinators. Sweet scents tend to attract bees and butterflies and moths, but some flies and beetles might prefer scents that signal fermentation or putrefaction. Flowers also provide protection for the ovule and developing embryo inside a receptacle. The function of the fruit is seed protection and dispersal. Different fruit structures or tissues on fruit, such as sweet flesh, wings, parachutes, or spines that grab, reflect the dispersal strategies that help spread seeds. Flowers are modified leaves or sporophylls, organized around a central receptacle. Although they vary greatly in appearance, virtually all flowers contain the same structures: sepals, petals, carpels, and stamens. The peduncle typically attaches the flower to the plant proper. A whorl of sepals, collectively called the calyx, is located at the base of the peduncle and encloses the unopened floral bud. Sepals are usually photosynthetic organs, although there are some exceptions. For example, the corolla in lilies and tulips consists of 3 sepals and 3 petals that look virtually identical. Petals, collectively the corolla, are located inside the whorl of sepals and may display vivid colors to attract pollinators. Sepals and petals together form the perianth. The sexual organs, the female gynoecium and male androecium, are located at the center of the flower. Typically, the sepals, petals, and stamens are attached to the receptacle at the base of the gynoecium, but the gynoecium may also be located deeper in the receptacle, with the other floral structures attached above it. As illustrated in Figure 26.14, the innermost part of a perfect flower is the gynoecium, the location in the flower where the eggs will form. The female reproductive unit consists of 1 or more carpels, each of which has a stigma, style, and ovary. The stigma is the location where the pollen is deposited either by wind or a pollinating arthropod. The sticky surface of the stigma traps pollen grains, and the style is a connecting structure through which the pollen tube will grow to reach the ovary. The ovary houses 1 or more ovules, each of which will ultimately develop into a seed. Flower structure is very diverse, and carpels may be singular, multiple, or fused. Multiple fused carpels comprise a pistil. The androecium, or male reproductive region, is composed of multiple stamens surrounding the central carpel. Stamens are composed of a thin stalk called a filament and a sac-like structure called the anther. The filament supports the anther, where the microspores are produced by meiosis and develop into haploid pollen grains, or male gametophytes. 26.3. Angiosperms. 681. Figure 26.14 Flower structure. This image depicts the structure of a perfect flower. Perfect flowers produce both male and female floral organs. The flower shown has only 1 carpel, but some flowers have a cluster of carpels. Together, all the carpels make up the gynoecium. Credit. Modification of work by Mariana Ruiz Villarreal. The life cycle of an angiosperm. The adult or sporophyte phase is the main phase of an angiosperm's life cycle, figure 26.15. Like gymnosperms, angiosperms are heterosporous. Therefore, they produce microspores, which will generate pollen grains as the male gametophytes, and megaspores, which will form an ovule that contains female gametophytes. Inside the anther's microsporangia, male sporocytes divide by meiosis to generate haploid microspores, which, in turn, undergo mitosis and give rise to pollen grains. Each pollen grain contains two cells, one generative cell that will divide into two sperm and a second cell that will become the pollen tube cell. 682 Chapter 26, Seed Plants access for free at openstax.org. Visual connection figure 26.15 Angiosperm life cycle. The life cycle of an angiosperm is shown. Anthers and carpels are structures that shelter the actual gametophytes, the pollen grain and embryo sac. Double fertilization is a process unique to angiosperms. Credit. Modification of work by Mariana Ruiz Villarreal. Question. If a flower lacked a megasporangium, what type of gamete would not form? If the flower lacked a microsporangium, what type of gamete would not form? The ovule, sheltered within the ovary of the carpel, contains the megasporangium protected by two layers of integuments and the ovary wall. Within each megasporangium, a diploid megasporocyte undergoes meiosis, generating 4 haploid megaspores, 3 small and 1 large. Only the large megaspore survives. It divides mitotically 3 times to produce 8 nuclei distributed among the 7 cells of the female gametophyte or embryo sac. 3 of these cells are located at each pole of the embryo sac. The 3 cells at one pole become the egg and 2 synergids. The 3 cells at the opposite pole become antipodal cells. The center cell contains the remaining 2 nuclei, polar nuclei. This cell will eventually produce the endosperm of the seed. The mature embryo sac then contains 1 egg cell, 2 synergids or helper cells, 3 antipodal cells, which eventually degenerate, and a central cell with 2 polar nuclei. When a pollen grain reaches the stigma, a pollen tube extends from the grain, grows down the style, and enters through the micropyle, an opening in the integuments of the ovule. The 2 sperm are deposited in the embryo sac. A double fertilization event then occurs. 1 sperm and the egg combine, forming a diploid zygote, the future embryo. The other sperm fuses with the polar nuclei, forming a triploid cell that will develop into the endosperm, the tissue that serves as a food reserve for the developing embryo. The zygote develops into an embryo with a radicle, or small root, and 1 monocot, or 26.3, angiosperms 683-2, dicot, leaf-like organs called cotyledons. This difference in the number of embryonic leaves is the basis for the 2 major groups of angiosperms, the monocots and the eudicots. Seed food reserves are stored outside the embryo, in the form of complex carbohydrates, lipids, or proteins. The cotyledons serve as conduits to transmit the broken-down food reserves from their storage site inside the seed to the developing embryo. The seed consists of a toughened layer of integuments forming the coat, the endosperm with food reserves, and at the center, the well-protected embryo. Most angiosperms have perfect flowers, which means that each flower carries both stamens and carpels, figure 26.15. In monoecious plants, male, staminate, and female, pistillate, flowers are separate, but carried on the same plant. Sweetgums, liquidambar spp, and beeches, Betula spp, are monoecious, figure 26.16. In dioecious plants, male and female flowers are found on separate plants. Willows, Salix spp, and poplars, Populus spp, are dioecious. In spite of the predominance of perfect flowers, only a few species of angiosperms self-pollinate. Both anatomical and environmental barriers promote cross-pollination mediated by a physical agent, wind or water, or an animal, such as an insect or bird. Cross-pollination increases genetic diversity in a species. Figure 26.16 Beech inflorescences. The female inflorescence is at the upper left. The male inflorescence is at the lower right. Credit. Stephen J. Baskow, 2002. http://bioimages.vanderbilt.edu/bascalf/10593 http://openstax.org/l/betula Morphbank. Biological imaging, http://www.morphbank.net/ http://openstax.org/l/morphbank 29th of June 2017. Florida State University, Department of Scientific Computing, Tallahassee, Florida, 323064026 USA. Fruit. As the seed develops, the walls of the ovary thicken and form the fruit. The seed forms in an ovary, which also enlarges as the seeds grow. Many foods commonly called vegetables are actually fruits. Eggplants, zucchini, string beans, tomatoes, and bell peppers are all technically fruits because they contain seeds and are derived from the thick ovary tissue. Acorns are true nuts, and winged maple, helicopter seeds, or whirligigs, whose botanical name is samara, are also fruits. Botanists classify fruit into more than 24 different categories, only a few of which are actually fleshy and sweet. Mature fruit can be fleshy or dry. Fleshy fruit include the familiar berries, peaches, apples, grapes, and tomatoes. Rice, wheat, and nuts are examples of dry fruit. Another subtle distinction is that not all fruits are derived from just the ovary. For instance, strawberries are derived from the ovary as well as the receptacle, and apples are formed from the ovary and the pericarp, or 684 Chapter 26. Seed plants access for free at openstax.org. Hypanthium. Some fruits are derived from separate ovaries in a single flower, such as the raspberry. Other fruits, such as the pineapple, form from clusters of flowers. Additionally, some fruits, like watermelon and orange, have rinds. Regardless of how they are formed, fruits are an agent of seed dispersal. The variety of shapes and characteristics reflect the mode of dispersal. Wind carries the light dry fruits of trees and dandelions. Water transports floating coconuts. Some fruits attract herbivores with their color or scent, or as food. Once eaten, tough, undigested seeds are dispersed through the herbivore's feces, endozoochory. Other fruits have burrs and hooks to cling to fur and hitch rides on animals, epizoochory. Diversity of angiosperms. Angiosperms are classified in a single phylum, the Anthophyta. Modern angiosperms appear to be a monophyletic group, which as you may recall means that they originated from a single ancestor. Within the angiosperms are three major groups: basal angiosperms, monocots, and dicots. Basal angiosperms are a group of plants that are believed to have branched off before the separation of the monocots and eudicots, because they exhibit traits from both groups. They are categorized separately in most classification schemes. The basal angiosperms include ambarella, water lilies, the magnoliids, magnolia trees, laurels, and spice peppers, and a group called the Austrobaleales, which includes the star anise. The monocots and dicots are differentiated on the basis of the structure of the cotyledons, pollen grains, and other structures. Monocots include grasses and lilies, and the dicots form a multi-branched group that includes, among many others, roses, cabbages, sunflowers, and mints. Basal angiosperms. The Magnoliidae are represented by the magnolias, laurels, and peppers. Magnolias are tall trees bearing dark, shiny leaves and large, fragrant flowers with many parts, and are considered archaic, figure 26.17. In the outer whorl of the magnolia flower the sepals and petals are undifferentiated and are collectively called tepals. The reproductive parts are arranged in a spiral around a cone-shaped receptacle, with the carpels located above the stamens, figure 26.17. The aggregate fruit, with one seed formed from each carpel, is seen in figure 26.18d. Laurel trees produce fragrant leaves and small, inconspicuous flowers. The laurels grow mostly in warmer climates and are small trees and shrubs. Familiar plants in this group include the bay laurel, cinnamon, spicebush, figure 26.18a, and avocado tree. Figure 26.17 Magnolia grandiflora. A cluster of carpels can be seen above the stamens, which have shed their pollen and begun to drop from the inflorescence. In the flower, the sepals and petals are undifferentiated and are collectively called tepals. Credit. Ianare Savi. http://bioimages.vanderbilt.edu/bascalf/109949 http://openstax.org/l/grandiflora (26.3) Angiosperms 685 Figure 26.18 Basal angiosperms. The common spicebush belongs to the laurels, the same family as cinnamon and bay laurel. The fruit of B. the Piper nigrum plant is black pepper, the main product that was traded along spice roots. Notice the small, unobtrusive, clustered flowers. The leaf venation resembles that of both the monocots, parallel, and the dicots, branched. C. Water lilies, Nymphaea lotus. Although the leaves of the plant float on the surface of the water, their roots are in the underlying soil at the bottom of the lake. The aggregate fruit of a magnolia, D. The fruit is in its final stage, with its red seeds just starting to appear. Credit A. Modification of work by Corey Zanker. Credit B. Modification of work by Franz Eugen Kohler. Credit C. Modification of work by R.L. Wikimedia Commons. D. Modification of work by Coastside 2 inches Wikimedia Commons. Monocots. Plants in the monocot group are primarily identified by the presence of a single cotyledon in the seedling. Other anatomical features shared by monocots include veins that run parallel to and along the length of the leaves, and flower parts that are arranged in a 3- or 6-fold symmetry. True woody tissue is rarely found in monocots. In palm trees, vascular and parenchyma tissues produced by the primary and secondary thickening meristems form the trunk. The pollen from the first angiosperms was likely monosulcate, containing a single furrow or pore through the outer layer. This feature is still seen in the modern monocots. Vascular tissue of the stem is scattered, not arranged in any particular pattern, but is organized in a ring in the roots. The root system consists of multiple fibrous roots, with no major taproot. Adventitious roots often emerge from the stem or leaves. The monocots include familiar plants such as the true lilies, Liliopsida, orchids, yucca, asparagus, grasses, and palms. Many important crops are monocots, such as rice and other cereals, corn, sugarcane, and tropical fruits like bananas and pineapples. Figure 26.19a, b, c. 686 Chapter 26, Seed Plants Access for Free at OpenStax.org. Figure 26.19 Monocot and dicot crop plants. The world's major crops are flowering plants. A. Rice, B. Wheat, and C. Bananas are monocots, while D. Cabbage, E. Beans, and F. Peaches are dicots. Credit A. Modification of work by David Nance, USDA Rs. Credit B. C. Modification of work by Rosendahl. Credit D. Modification of work by Bill Tarpening, USDA. Credit E. Modification of work by Scott Bauer, USDA Rs. Credit F. Modification of work by Keith Weller, USDA. Eudicots, or true dicots, are characterized by the presence of two cotyledons in the developing shoot. Veins form a network in leaves, and flower parts come in 4, 5, or many whorls. Vascular tissue forms a ring in the stem. In monocots, vascular tissue is scattered in the stem. Eudicots can be herbaceous, not woody, or produce woody tissues. Most eudicots produce pollen that is trisulcate or triparate, with 3 furrows or pores. The root system is usually anchored by one main root developed from the embryonic radicle. Eudicots comprise 2/3 of all flowering plants. The major differences between monocots and eudicots are summarized in Table 26.1. However, some species may exhibit characteristics usually associated with the other group, so identification of a plant as a monocot or a eudicot is not always straightforward. Comparison of structural characteristics of monocots and eudicots. Characteristic monocot-eudicot cotyledon 1-2 veins in leaves parallel network. Branched, stem vascular tissue scattered arranged in ring pattern roots network of fibrous roots taproot with many lateral roots pollen monosulcate trisulcate flower parts 3 or multiple of 3 4, 5, multiple of 4 or 5 and whorls. Table 26.1. 26.4 The role of seed plants. By the end of this section, you will be able to do the following. Explain how angiosperm diversity is due, in part, to multiple complex interactions with animals. Describe ways in which pollination occurs. Discuss the roles that plants play in ecosystems and how deforestation threatens plant biodiversity. Without seed plants, life as we know it would not be possible. Plants play a key role in the maintenance of terrestrial ecosystems through the stabilization of soils, cycling of carbon, and climate moderation. Large tropical forests release oxygen and act as carbon dioxide sinks. Seed plants provide shelter to many life forms, as well as food for herbivores, thereby indirectly feeding carnivores. Plant secondary metabolites are used for medicinal purposes and industrial production. Virtually all animal life is dependent on plants for survival. Animals and plants. Herbivora coevolution of flowering plants and insects is a hypothesis that has received much attention and support, especially because both angiosperms and insects diversified at about the same time in the Middle Mesozoic. Many authors have attributed the diversity of plants and insects to both pollination and herbivora, or the consumption of plants by insects and other animals. Herbivora is believed to have been as much a driving force as pollination. Coevolution of herbivores and plant defenses is easily and commonly observed in nature. Unlike animals, most plants cannot outrun predators or use mimicry to hide from hungry animals, although mimicry has been used to entice pollinators. A sort of arms race exists between plants and herbivores. To combat herbivores, some plant seeds, such as acorn and unripened persimmon, are high in alkaloids and therefore unsavory to some animals. Other plants are protected by bark, although some animals develop specialized mouthpieces to tear and chew vegetal material. Spines and thorns, figure 26.20, deter most animals, except for mammals with thick fur, and some birds have specialized beaks to get past such defenses. Figure 26.20 Plant defenses. A. Spines and B. Thorns are examples of plant defenses. Credit A. Modification of work by John Sullivan. Credit B. Modification of work by I. Sasek Sr. Herbivora has been exploited by seed plants for their own benefit. The dispersal of fruits by herbivorous animals is a striking example of mutualistic relationships. The plant offers to the herbivore a nutritious source of food in return for spreading the plant's genetic material to a wider area. An extreme example of coevolution, discovered by Daniel Janssen, between an animal and a plant is exemplified by Mexican acacia trees and their attendant acacia ants Pseudomyrmex spp. This is termed myrmecophytism. The trees support the ants with shelter and food. The ants nest in the hollows of large thorns produced by the tree and feed on sugary secretions produced at the ends of the leaves. The sugar pellets also help to keep the ants from interfering with insect pollinators. In return, ants discourage herbivores, both invertebrates and vertebrates, by stinging and attacking leaf eaters and insects ovipositing on the plants. The ants also help to remove potential plant pathogens, such as fungal growths. Another case of insect-plant coevolution is found in bracken fern, Pteridium equinolum, whose subspecies are found throughout the world. Bracken ferns produce a number of secondary plant compounds in their adult fronds that serve as defensive compounds against non-adapted insect attack. These compounds include cyanogenic glucosides, tannins, and phenolics. However, during the fiddlehead or crozier stage, bracken secretes nutritious sugary and proteinaceous compounds from special nectaries that attract ants and even species of jumping spiders, all of which defend the plant's croziers until they are fully unfolded. These opportunistic groups of protective arthropods greatly reduce the damage that otherwise would occur during the early stages of growth. 688 Chapter 26, Seed Plants Access for Free at OpenStax.org. Animals and plants. Pollination. Flowers pollinated by wind are usually small, feathery, and visually inconspicuous. Grasses are a successful group of flowering plants that are wind-pollinated. They produce large amounts of powdery pollen carried over large distances by the wind. Some large trees such as oaks, maples, and birches are also wind-pollinated. Link to learning. Explore this website, http://openstax.org/l/pollinators2 for additional information on pollinators. More than 80% of angiosperms depend on animals for pollination, technically the transfer of pollen from the anther to the stigma. Consequently, plants have developed many adaptations to attract pollinators. With over 200,000 different plants dependent on animal pollination, the plant needs to advertise to its pollinators with some specificity. The specificity of specialized plant structures that target animals can be very surprising. It is possible, for example, to determine the general type of pollinators favored by a plant by observing the flower's physical characteristics. Many bird or insect-pollinated flowers secrete nectar, which is a sugary liquid. They also produce both fertile pollen, for reproduction, and sterile pollen rich in nutrients for birds and insects. Many butterflies and bees can detect ultraviolet light, and flowers that attract these pollinators usually display a pattern of ultraviolet reflectance that helps them quickly locate the flower center. In this manner, pollinating insects collect nectar while at the same time are dusted with pollen. Figure 26.21. Large, red flowers with little smell and a long funnel shape are preferred by hummingbirds, who have good color perception, a poor sense of smell, and need a strong perch. White flowers that open at night attract moths. Other animals, such as bats, lemurs, and lizards, can also act as pollinating agents. Any disruption to these interactions, such as the disappearance of bees, for example as a consequence of colony collapse disorders, can lead to disaster for agricultural industries that depend heavily on pollinated crops. Figure 26.21 Pollination. As a bee collects nectar from a flower, it is dusted by pollen, which it then disperses to other flowers. Credit: John Severn's Scientific Method Connection Testing Attraction of Flies by Rotting Flesh Smell Question. Will flowers that offer cues to bees attract carrion flies if sprayed with compounds that smell like rotten flesh? Background: visitation of flowers by pollinating flies is a function mostly of smell. Flies are attracted by rotting flesh and carrions. The putrid odor seems to be the major attractant. The polyamines putrescine and cadaverine, which are the products of protein breakdown after animal death, are the source of the pungent smell of decaying meat. Some plants strategically attract flies by synthesizing polyamines similar to those generated by decaying flesh and thereby attract carrion flies. Flies seek out dead animals because they normally lay their eggs on them and their maggots feed on the decaying flesh. Interestingly, time of death can be determined by a forensic entomologist based on the stages and type of maggots recovered from cadavers. 26.4. The role of seed plants. 689 Hypothesis. Because flies are drawn to other organisms based on smell and not sight, a flower that is normally attractive to bees because of its colors will attract flies if it is sprayed with polyamines similar to those generated by decaying flesh. Test the hypothesis. 1. Select flowers usually pollinated by bees. White petunia may be a good choice. 2. Divide the flowers into two groups, and while wearing eye protection and gloves, spray one group with a solution of either putrescine or cadaverine. Putrescine dihydrochloride is typically available in 98% concentration. This can be diluted to approximately 50% for this experiment. 3. Place the flowers in a location where flies are present, keeping the sprayed and unsprayed flowers separated. 4. Observe the movement of the flies for 1 hour. Record the number of visits to the flowers using a table similar to Table 26.2. Given the rapid movement of flies, it may be beneficial to use a video camera to record the fly-flower interaction. Replay the video in slow motion to obtain an accurate record of the number of fly visits to the flowers. 5. Repeat the experiment 4 more times with the same species of flower, but using different specimens. 6. Repeat the entire experiment with a different type of flower that is normally pollinated by bees. Results of number of visits by flies to sprayed and control, unsprayed flowers trial # sprayed flowers unsprayed flowers 1 2 3 4 5 Table 26.2 Analyze your data. Review the data you have recorded. Average the number of visits that flies made to sprayed flowers over the course of the 5 trials, on the first flower type, and compare and contrast them to the average number of visits that flies made to the unsprayed control flowers. Can you draw any conclusions regarding the attraction of the flies to the sprayed flowers? For the second flower type used, average the number of visits that flies made to sprayed flowers over the course of the 5 trials and compare and contrast them to the average number of visits that flies made to the unsprayed control flowers. Can you draw any conclusions regarding the attraction of the flies to the sprayed flowers? Compare and contrast the average number of visits that flies made to the 2 flower types. Can you draw any conclusions about whether the appearance of the flower had any impact on the attraction of flies? Did smell override any appearance differences, or were the flies attracted to one flower type more than another? Form a conclusion. Do the results support the hypothesis? If not, how can your observations be explained? The importance of seed plants in human life. Seed plants are the foundation of human diets across the world. Figure 26.22. Many societies eat almost exclusively vegetarian fare and depend solely on seed plants for their nutritional needs. A few crops, rice, wheat, and potatoes, dominate the agricultural landscape. Many crops were developed during the agricultural revolution, when human societies made the transition from nomadic hunter-gatherers to horticulture and agriculture. Cereals, rich in carbohydrates, provide the staple of many human diets. Beans and nuts supply proteins. Fats are derived from crushed seeds, as is the case for peanut and rapeseed, canola, oils, or fruits such as olives. Animal husbandry also consumes large quantities of crop plants. Staple crops are not the only food derived from seed plants. Various fruits and vegetables provide nutrient macromolecules. 690 Chapter 26. Seed plants access for free at OpenStax.org. Vitamins, minerals, and fiber. Sugar, to sweeten dishes, is produced from the monocot sugarcane and the eudicot sugar beet. Drinks are made from infusions of tea leaves, chamomile flowers, crushed coffee beans, or powdered cocoa beans. Spices come from many different plant parts. Saffron and cloves are stamens and buds. Black pepper and vanilla are seeds. The bark of a bush in the laurels family supplies cinnamon, and the herbs that flavor many dishes come from dried leaves and fruit, such as the pungent red chili pepper. The volatile oils of a number of flowers and bark provide the scent of perfumes. Additionally, no discussion of seed plant contribution to human diet would be complete without the mention of alcohol. Fermentation of plant-derived sugars and starches is used to produce alcoholic beverages in all societies. In some cases, the beverages are derived from the fermentation of sugars from fruit, as with wines, and, in other cases, from the fermentation of carbohydrates derived from seeds, as with beers. The sharing of foods and beverages also contributes to human social ritual. Seed plants have many other uses, including providing wood as a source of timber for construction, fuel, and material to build furniture. Most paper is derived from the pulp of coniferous trees. Fibers of seed plants such as cotton, flax, and hemp are woven into cloth. Textile dyes, such as indigo, were mostly of plant origin until the advent of synthetic chemical dyes. Lastly, it is more difficult to quantify the benefits of ornamental seed plants. These grace private and public spaces, adding beauty and serenity to human lives and inspiring painters and poets alike. Figure 26.22 Human uses of plants. Humans rely on plants for a variety of reasons. A. Cacao beans were introduced to Europe from the New World, where they were used by Mesoamerican civilizations. Combined with sugar, another plant product, chocolate is a popular food. B. Flowers like the tulip are cultivated for their beauty. C. Quinine, extracted from cinchona trees, is used to treat malaria, to reduce fever, and to alleviate pain. D. This violin is made of wood. Credit A. Modification of work by Evergene Flicker. Credit B. Modification of work by Rosendahl. Credit C. Modification of work by Franz Eugen Kohler. 26.4. The role of seed plants. 691. The medicinal properties of plants have been known to human societies since ancient times. There are references to the use of plants' curative properties in Egyptian, Babylonian, and Chinese writings from 5,000 years ago. Many modern synthetic therapeutic drugs are derived or synthesized de novo from plant secondary metabolites. It is important to note that the same plant extract can be a therapeutic remedy at low concentrations, become an addictive drug at higher doses, and can potentially kill at high concentrations. Table 26.3 presents a few drugs, their plants of origin, and their medicinal applications. Plant origin of medicinal compounds and medical applications. Plant compound application. Deadly nightshade, Atropa belladonna, atropine. Dilate eye pupils for eye exams. Foxglove, Digitalis purpurea, digitalis heart disease, stimulates heartbeat. Yam, Dioscorea spp. Steroids. Steroid hormones, contraceptive pill and cortisone. Ephedra, ephedra spp. Ephedrine decongestant and bronchiole dilator. Pacific U, Taxus brevifolia, Taxol. Cancer chemotherapy, inhibits mitosis. Opium poppy, Papaver somniferum, opioids. Analgesic, reduces pain without loss of consciousness, and narcotic, reduces pain with drowsiness and loss of consciousness. In higher doses, quinine tree, cinchona spp. Quinine antipyretic, lowers body temperature, and antimalarial willow, salix spp. Salicylic acid, aspirin, analgesic and antipyretic. Table 26.3. Career connection. Ethnobotanist. The relatively new field of ethnobotany studies the interaction between a particular culture and the plants native to the region. Seed plants have a large influence on day-to-day human life. Not only are plants the major source of food and medicine, they also influence many other aspects of society, from clothing to industry. The medicinal properties of plants were recognized early on in human cultures. From the mid-1900s, synthetic chemicals began to supplant plant-based remedies. Pharmacognosy is the branch of pharmacology that focuses on medicines derived from natural sources. With massive globalization and industrialization, it is possible that much human knowledge of plants and their medicinal purposes will disappear with the cultures that fostered them. This is where ethnobotanists come in. To learn about and understand the use of plants in a particular culture, an ethnobotanist must bring in knowledge of plant life and an understanding and appreciation of diverse cultures and traditions. The Amazon forest is home to an incredible diversity of vegetation and is considered an untapped resource of medicinal plants. Yet, both the ecosystem and its indigenous cultures are threatened with extinction. To become an ethnobotanist, a person must acquire a broad knowledge of plant biology, ecology, and sociology. Not only are the 692 Chapter 26 seed plants accessed for free at OpenStax.org. Plant specimens studied and collected, but also the stories, recipes, and traditions that are linked to them. For ethnobotanists, plants are not viewed solely as biological organisms to be studied in a laboratory, but as an integral part of human culture. The convergence of molecular biology, anthropology, and ecology make the field of ethnobotany a truly multidisciplinary science. Biodiversity of plants. Biodiversity ensures a resource for new food crops and medicines. Plant life balances ecosystems, protects watersheds, mitigates erosion, moderates our climate, and provides shelter for many animal species. Threats to plant diversity, however, come from many sources. The explosion of the human population, especially in tropical countries where birth rates are highest and economic development is in full swing, is leading to devastating human encroachment into forested areas. To feed the growing population, humans need to obtain arable land, so there has been and continues to be massive clearing of trees. The need for more energy to power larger cities and economic growth therein leads to the construction of dams, the consequent flooding of ecosystems, and increased emissions of pollutants. Other threats to tropical forests come from poachers, who log trees for their precious wood. Ebony and Brazilian rosewood, both on the endangered list, are examples of tree species driven almost to extinction by indiscriminate logging. This unfortunate practice continues unabated today, largely due to lack of population control and political willpower. The number of plant species becoming extinct is increasing at an alarming rate. Because ecosystems are in a delicate balance, and seed plants maintain close symbiotic relationships with animals, whether predators or pollinators, the disappearance of a single plant can lead to the extinction of connected animal species. A real and pressing issue is that many plant species have not yet been catalogued, and so their place in the ecosystem is unknown. These unknown species are threatened by logging, habitat destruction, and loss of pollinators. They may become extinct before we have the chance to begin to understand the possible impacts from their disappearance. Efforts to preserve biodiversity take several lines of action, from preserving heirloom seeds to barcoding species. Heirloom seeds come from plants that were traditionally grown in human populations, as opposed to the seeds used for large-scale agricultural production. Barcoding is a technique in which one or more short gene sequences, taken from a well-characterized portion of DNA found in most genomes, are used to identify a species through DNA analysis.